This is a production of Cornell University.
Yeah, so thanks, Luke, for the introduction. And yeah, so I want to share some of what I've been doing and listening to Luke and preparing for this. If you sum up everything I did for a short period of time, it seems like I've done a lot of things, and that's a really nice thing to reflect on. So I did make this plan for what I wanted to touch on today. Um, I've, I've, I prepared for this and I ran this a bunch of times, so I can either go really quickly and be way too quick, or I might be a little bit over, so I'll try and be cognizant of time. I apologize in advance for any discrepancies, but what I want to do is I want to talk a little bit about how I got to do what I'm doing and some of the stuff I did during my PhD, which was working on TEF, and then how that led to plant-plant interactions, and then working on stuff we've done recently, things we are doing, and things we hope to do. And yeah, I'm gonna hope to leave a lot of time for questions. So far, everyone I've interacted with here has been both kind and smart, so I really want to hear what you all have to say. So I grew up there. I've been working on a farm since I was 11. I've been driving tractors for a very long time. And this was, I guess, pretty diverse. I've worked on multiple crops, but working on crops is actually not where my interest in plants came from. I fell in love with plants here. These are meadows that are very close to my home, where I would hike as a child, and this is one of my favorite places in the world. And when you look at where I got interested in plants, you can see just how many plants grow here together. So in a sharp contrast to the monoculture you see behind of grapes and almonds and olives and all that, you have legumes here and cereals here and a whole bunch of other genres and families all growing together, and they make it work. And I think that's what makes them, as much as us, what makes them appealing is how diverse they are, how interesting they are, and how they get along. But there really wasn't a study the meadows behind your house class, so I went into crop science instead. And so I started learning about TEF as an undergrad, working in Professor Yoshua Saranga's lab at the Hebrew University. And very early on, it piqued my interest because I found out that teff has a very large demand in Israel, which is not where it's endemic to. Teff is a cereal crop that's endemic to Ethiopia, if any of you are unfamiliar. And in the early 90s, there was a big wave of repatriotism of Jews who came from Ethiopia to Israel, and they continued to consume teff as their staple. Being recent immigrants, this wasn't the best well-off population, and at the same time, the cost they paid for the staple flour was 3 times as high as that of wheat flour, which to us, it made us scream to high heavens, and we got interested in lowering the price by producing it locally. And on top of that, farmers in Israel, as well as here, always needed more crops in the rotation. We always need more things to grow. We need the diversity. We need those changes. And there was a specific need for a short season cereal crop that can utilize excess nutrients and water from the soil for specific farmers, specifically those who grew carrots and potatoes. They had a large interest in teff. So with all that in mind, we said, hey, let's work on it. Let's see if we can grow teff in Israel. And starting off, we had some ideas of how teff grew, but we had a lot of questions. And all of these were non-obvious when we started. So we were going off of some ideas we got from people who came from Ethiopia and were farmers previously. And I spent substantial amounts of time trying to adapt practices used in smallholder traditional farming in Ethiopia to highly mechanized, irrigated agriculture like the type we have. And we had to answer each of this. So literally, which planter to use for these tiny seeds. when to sow, how to irrigate. Do we use sprinklers or pivots? How do we even get the right yields? What sort of genetics we needed for those yields? And then later on, we focused on lodging, which I'm going to touch on heavily in this. And then we also had the question of how to harvest, which actually became the easiest one to answer. I went on YouTube, I Googled TEF, and we found a video from Idaho, and slam dunk. I wish everything was that easy. But everything else we had to calibrate. So we literally had to ask that question. And so we did get some answers. I won't go into the whole process of this, but we did find that if we sowed in early spring, we got up to 150% more yield compared to some other periods throughout the time. So the timing was crucial. And then we also found out that, as luck would have it, there was a planter that was heavily sold back home in the 80s, a Great Plains planter that had a small seed chamber in it that fit TEF very, very good. And it was pretty accurate in sowing densities, which you'll soon realize is very important as well. We also tested what the right density of plants was, and we found that that produced really high increases in yield as well. And TEF seeds being this big, which is about between a fifth or a third of a sesame seed, so teeny tiny, we had to find out if we can sow them or actually cover them with soil and not put them on the soil surface, which was what farmers were doing at the time, since they looked at these plants and said, this is never going to make it out of the soil. But we actually found out that they can make it from pretty substantial depth, and that if we covered them, we decreased seed desiccation, so we got better establishment, and we got better anchorage. So the roots, the shoot-to-root junction happened below the ground, and that improved the plant anchorage, which is a major determinant in lodging. And as for the genetics and how to get the best yields, I feel like it's like I won the lottery. There's a lot of luck involved in this, as with any science, but we wanted to work on TEF. We needed genetics. We reached out to Ethiopia. It was made clear to us that that's never going to happen, and not a single seed was going to leave their germplasm to come to us. So then we looked locally, and we went to a conference. We spoke to someone who worked at the Israeli Gene Bank and said, hey, but we have TEF. And then we found out that we actually had a collection that was collected by the USDA in the 50s and 60s, brought here. Something was done to it, and then for whatever reason, it was shipped to Israel to be shared with Israeli researchers, and basically disappeared. We don't know who ordered it, who got it, who maintained it. We knew nothing. So we started with those. Some were pretty obvious mixtures of different colors of seeds, and we had to sort of stabilize them genetically. We did a single seed descent stabilizing phase and created a panel of just under 300 lines. We then used that panel for two things. One, we tested it in a bunch of different locations, trying to see if anything stands out, and if we can see a line that's readily adaptable to Israeli agriculture, which I'll share. I've said it to some of you this morning. I just found out that one of the lines we started licensing is entered the final stage, and not only that, but it's the line that's named after my son. So that's pretty cool. And so we used that panel to do a GBS. We then used that GBS data to do a preliminary GWAS and try and map key traits to specific loci. And we were able to find hits for a lot of important traits. But the really cool thing we found is that multiple sites were shared between the three biggest things we were targeting. So days to heading, grain yield, and lodging index all shared specific locations on the genome, which was a very exciting find. Both Muluken, who worked on this with me, and I have since finished and moved to something else, but I'm really hoping somebody picks this up because I think it's really interesting. Yeah, and then the big issue we were dealing with is lodging, which is what you see here. And what I'm about to say is somewhat contested in the TEF science ecosystem, so I hope nobody gets offended. Lodging, generally, is the permanent displacement of a plant from the upright position. That part is not contested. What is contested is if this is root lodging or shoot lodging. So in the literature, root lodging is a partial uprooting, which I think that's what we're seeing here. And shoot lodging is a bending of the shoot, which also happens, except it happens after this. So I'm going to say it's root lodging. If you're contested, I agree with you. And then it's a big issue. So compared to a three-celled or supported growth, this leads to up to 50% yield reduction in both biomass and grain yield. And this environment that's created here is the perfect place for bugs and fungus and pathogens of all natures. It's a big, big issue. And to make things worse, it makes it really hard for the harvester to go over it because those plants just stuck under. This has been possibly less of an issue in Ethiopia where the most of the harvesting is still manual. But it's still, all of those losses still apply there as well. So we saw this, and I was a TA at the time, which I loved. And I was a TA for Intro to Plant Science. And one of the things we got to talk about is shoot development and the effects that shading has on things. So we said, hey, maybe, TA brain at the time, maybe the plants are sensing shading, thus they are becoming taller, thus they're losing their anchorage because the center of gravity rises. Maybe that's why they topple over. So we said, okay, what causes this? And possibly higher densities, right? It's a pretty short jump in my brain. And then we tested. So as a context to this, when we started out, the data we had was that people are sowing up to 20 kilograms per hectare, which is a lot. The seeds are tiny. We're talking billions of plants per a few meters. It was very dense. So we said, okay, this is probably too much. Let's cut it. So we went to 9 kilograms, which is about half, all the way down to 3 kilograms per hectare. I'm sorry for stumbling. We don't really use hectares in Israel. We use dunams because hectares are way too big and the country is way too small. So, but when we did this, we found that the plants grown in lower densities were taller, not shorter. So it didn't seem to be that effect. Also, we found that lodging was reduced, and that got me interested, and I was trying to yank those plants out, and I also found that the plants here just held on to the ground more. It's called anchorage, and that pointed to another thing that pointed us in the direction of root lodging. So that I felt was interesting, and then we got to thinking, what in the root could it be? And this is about the time that Jonathan Lynch, who at the time I was afraid of, now I know him, he's actually very nice. He had a paper about root cortical parenchyma, and that got us thinking about maybe that's what's happening. Maybe some of the areas of the cortex are deforming, and then the connection between the side that's attached to the soil and the side that is definitely connected to the shoot, maybe something there breaks. And when we looked at it, we saw that some of the more lodging resistant lines, and they're not resistant completely, but more so, they produced less root cortical parenchyma, and they also produced it, later in the season. So they had improved retention of the cortex. What we think is happening is that with the partial uprooting, there's a significant secondary uprooting that's happening inside the root. So the steel could be just moving inside, much like a fiber moves inside an electrical wire, because it's not connected, and that could enable some flexibility to enable the plant to switch without disconnecting, because we're not seeing the roots cut, but we do see the plant lying on its side. So that could be, and I'm really happy about this. This was a simple light microscope. I was lucky enough to know the right people, and I can show you what it looks like under a scanning electron microscope, which was done by other people in our group. And that second work also found that other metrics of the root cortical area changed, but it seems to be the case. It seems to be that if a plant tends to lodge, that root cortical parenchyma is developed more and vice versa. So we don't know what starts the process, but there's definitely something going on with root anatomy when it comes to lodging. And because of what we knew about the densities and the changes we found based on densities, that got me thinking, how are plants telling one another what's going on? How are plants picking up signals that make them change their anatomy and their development, resulting in lodging or other metrics? And that's really how I landed in plant-plant interactions, which is sort of the in-between stage between my PhD and my postdoc. And so this is not new to you. Plants grow in diverse communities. If you don't believe me, there. But also under cultivation. So polycultures have been used for thousands of years. You all know this three sisters garden idea. It's amazing. It's an embodiment of symbiosis, right? Because the beans grow on top of the maize, getting support. They fixate nitrogen as a way of repaying the corn, is the way it's depicted at least. And then the squash grows and covers the soil, taking advantage of excess radiation. So we know this happens, and we know it works sustainably. But for all we know about plant-plant interaction, the below-ground part is less well known. And I can tell you why, because it's really hard to study. It's a lot of hard work, and it's the invisible part, right? And I'll spoil the rest of this, but I also focused on root exudates, which is the invisible part of the invisible part. So it's hard to study, but it holds a tremendous benefit potential because there's a big emphasis on soil, plant soil and plant microbe interactions. And those plant-plant interactions, specifically in intra-species ones, they cloud our view. They produce noise to other questions that we're asking. And so I think that's important. I was lucky enough to get to know Jesse Lasky, who also thought that was important. And I won a Bard Fellowship to go and get my postdoc with him. That has been the previous 2 years. And pretty short time before I came, this paper came out from the Lasky lab, which showed that in Arabidopsis plants, if all other things being equal and no shortage of nutrients, no shortage of water, the presence of competing plants, inter and intraspecies, caused changes to development. And in this specific paper, they were also able to mimic it by using a stragolactone standard and got similar responses. So we had an idea that the root exudates played a role here. And I was amazed at this. It's a whole new topic. And I spent some time thinking about how plants communicate with one another, excuse the very, I'm not a good drawer, I'm not good at drawing and less so on a computer. But we can think of some channels of plants communicating. We talked about shading. There's volatiles above ground. There's stuff that plants exude towards one another. Of course, there are microbes that are either shared or are used to transfer cues, sorry. And of course, we can talk about mycorrhizae. These are all things that we know. I can also say that I knew this was important because we know that agriculture is becoming ever more intensified. So we know plants are grown in higher and higher densities. And it is my hope that with regenerative agriculture and with newer, more eco-friendly systems, we're going to mix some crops at the same time. So intercropping became something that I was interested in as well. And the other question I had, being that I worked with plant selection and breeding before, was, is this breedable? Is this something we can breed for? And this is not a new idea. Since at least the late 60s, people have been talking about doing the selection of new lines under higher densities because agriculture was expected to keep on intensifying. And you probably know about the leaf angle changes in corn that make the plants fit better together and share more of the sunlight. But these are all above ground. Below ground traits that could benefit higher densities were less well documented at the time. So that was something we were very interested in. And we also thought that because seeds are sown very close to one another and they take longer to shade one another than to start exuding towards one another, probably root-to-root sensing happens before shading. So to try and check what the effect of this was, and there was a paper that showed that you can even transfer flowering signals through root exudates. So there was definitely enough evidence to support working on the communication below ground and trying to remove that from the context of above ground interactions. So we tested 10 sorghum lines. Some were land races, some were elite lines, all of which we grew under high fertilizer abundance with either drought conditions or well water conditions. And we did not allow plants to shade one another. What we did was we had focal plants of the 10 genotypes we tested, and we grew them in pots encircled by multiple plants or a single genotype that was the same competitor for all of these 10 lines. So one competitor competing against 10 lines. And we at first used these plates to push the plants to the side. The plants lifted the plate like with no issue at all. Then we tied them off to remove the leaves from being able to interact with one another. And that did produce stress, but we didn't see really signs of stress of those competitor plants. And then we asked the question, how does this affect the productivity and the development of plants? And what we found was that the presence of competition, which is the one on the second from the right, produced a smaller plant, reduced biomass, reduced the plant height, and reduced the leaf area. And that was on par with the control of a non-competition. This is supposed to be non-competition. A non-competition drought. So drought and the presence of neighbors led to similar reductions in productivity in sorghum. And this is intra-species. So a sorghum plant, sensing neighbors around it, reduced its productivity almost like it was experiencing drought. And then we looked at photosynthesis measurements of stomatal conductance and PS2 activity, and we saw a reduction in that as well. So not only is it smaller, it's producing less, it's less productive. And this could be, I can put this in a very kind context of saying it's just trying to share. It's trying not to steal everything. It's making itself small as a sort of survival through conservation. Or it could be that there's something going on there that's reducing its activity, similar to what we found in Arabidopsis before. So we saw that below ground interactions led to this reduction. And we said, okay, so if it's below ground, there's multiple things we can think of. It could be removal of nutrients on a very micro scale, so close to the root. Maybe that's what's going on. Or maybe it's a biochemical thing. Maybe it's an exudate thing. The plants are exuding something that's causing other plants to reduce their activity. And if that's the case, then we use this very high-tech system of a bucket and a funnel and a tube, where we grew source plants separately in a pot. We collected the leachate from this pot that had all the rhizo deposits from the same competitor genotype, and we transferred it to a target pot that either had actual competition in addition to this leachate or just this leachate. And we were wondering is this Q transported by exudates? And if the nature of that interaction will be different. The answers are very straightforward. Yes, there is a signal that's carried by exudates, and no, it's not different in nature. It seems to be the same thing. And what we found here was that the presence of the leachate of competing plants was similar in its effect to physical competition, plants grown in the same pot. We also found something that is very important for my current response to reviewers, where we did not see a heightened response when we had both competition and leachate. Because some of you are probably thinking allelopathy and ototoxicity. Ototoxicity, when it is created, produces a linear response across almost all concentrations. So this would have been here if this was a natural ototoxicity effect. And I'm a little bit bitter about it because I had paper reject. And so that was one thing we did. And working with sorghum, I was interested in what it did to other plants. And there are two reasons for this. One is when you dive into the literature about sorghum, there is a ton of papers saying that if you grow sorghum, the weed population is decreased, which didn't sit well with my experience of seeing weeds in sorghum fields, but I was interested in it. And then another thing was that we did a lot of work in Jesse Lasky's lab about working with striga and sorghum striga interactions. Striga is a parasitic plant in sub-Saharan Africa that picks on sorghum plants, and it's a major yield reducing element. And we were working on it. And one of the things in the process of working on it was getting root exudates to germinate Striga seeds. So we had that system, and I was wondering, and I was lucky enough to know Dr. Caroline Lowry, who worked with me on this, if we could see an effect, this plant-plant interaction between the root exudates that work so immensely on Striga seeds on some key weeds. So both common water hemp and foxtail this is giant foxtail, are major weeds in Pennsylvania. And we looked at germination, and we found, yes, the plants that were 5 to 14 days old, we grew them in nutrient solution. We then transferred them into DI water for 3 days, let them exude, and we used that to germinate seeds. And we found that those exudates reduced germination of both these species. So both foxtail and common water hemp were less germinated at a lesser extent when exposed to these exudates. And we were wondering if there was a killer agent in this. Is this a thing where the seeds just die? There's a toxic element to it. So we checked for seed viability, and it wasn't. And so with us seeing no reduction in seed viability, what we think is happening is that there is a secondary induced dormancy that's happening in the seeds, which seems to go along with gap sensing, which is a thing that small seeded weeds are known for, that if they see the light, in parentheses, but if they sense the sunlight, and if there's a niche that's available for them to occupy, they will germinate more. And perhaps what we're seeing is that those exudates serve as another agent sending the message of this area is taking. There is no gap. So this is what we're thinking. And we also looked at early development, and we saw in common water hemp, we didn't see any effect because the seeds were so tiny. We need a smaller ruler to test this. It's just, it was all within noise. But we found something interesting, that with giant foxtail, the seeds that did germinate developed much quicker. So they elongated much more. They produced a higher root mass when they were exposed to these exudates. How am I doing on time? So I'm going to run a little bit through this. But all of this work on sorghum led us and my reviewers to talk about allelopathy, which I dove in. I jumped into that rabbit hole. And I can say that the original paper, the seminal paper for allelopathy, says that allelopathy is any interaction between one plant and another, not necessarily deleterious. And with that, yes, what I'm seeing here is allelopathy. It's just not ototoxicity, not allelopathy in the common way that it's spoken. You'll excuse the football joke. And with this in mind and these papers being written, I started a new postdoc working on intercropping, with the understanding that with newer practices coming along, we're going to need intercropping to work. And the benefits of intercropping, depending on who you read, are immense. Anything from nutrient availability going up to pest resistance going up to higher yields, they're all presented as possible. And for us, they fit another idea that my current advisor, Dr. Guoji Wang, has in mind, where if we convinced Pennsylvania farmers who are growing silage to replace 10% of their corn plants with soy and sorghum, we can get a higher protein content in the silage. We can get a higher sugar content, making the in-sealing process quicker, without substantial yield reduction, making a better product quicker for the cows to eat. And the secondary effect that we are also very interested in is because there's a lot of dairy farms and a lot of cows producing a lot of manure, that manure gets put on the fields. The farmers then get fined if there's leaching of nitrogen or runoff of phosphorus, because it leads into the Chesapeake Bay and causes a lot of troubles. And there are evidence in the literature where if you grow this, you get higher nutrient uptake. And so this is why we're interested in this. And we got to thinking about those three crops. And a lot of everything I'm going to present in the next couple of minutes was done by these three women, Risha over there and Marin and Deirdre, and they did basic things. They're undergrads, and I'm very lucky to have had them work with me. And we were wondering, in planting these, how much longer do I have?
If you want to leave time for questions, we should stop around 1.
Oh, perfect. So I can slow down. So we got to thinking about how those fields of intercropping might look like and what the practices might be. And to me, it was very obvious that we should tell the farmers, plant the soy and sorghum first. Let them establish their slower growing. You Americans, you did a great job with your corn. It grows so quickly. I was amazed the first couple of times, but not the same effort has been done on soy and sorghum. And so I said, let's plant those first, let them establish, and then put in the corn. But I was assured that there's no way the farmers are going to go twice. There's just no chance. So basically, we knew that soy and sorghum are going to be germinating in an area that has corn plants that are bigger and already exuding. So we got thinking, what would the effect be? What would the exudates do in terms of early development, which we did in these germination papers, and to actual germination, which we did in petri dishes. So we grew these, all 3 species in a hydroponic system. We then transferred the exudates to plates or to these tubes to see what the effect was. And this is what the seedlings look like in germination paper, and this is what we found. So we saw that for corn, any sort of exudates that you put next to the seeds reduced its germination rate. So there is a heightened sensitivity there. We also found that sorghum does not care. It will just germinate. And we found that with soy, it's more picky. So with corn and sorghum, there was a reduction in germination rate. With soy exudates, soy seeds germinating in soy exudates, no substantial reduction. And we also looked at germinating dynamics. So this is a hydrothermal timeline model, which we applied to the germination data. And what we found was the water was constantly the fastest to induce germination. We did not find any effect for corn between soy and corn, and we saw a reduction in sorghum for all 3 of them. So it germinated less and slower. So there was an effect, but it could be, by the way, that slower effect is just the decomposing of the exudates. We don't know how long some of the compounds in the exudates live for. We have not done that metabolomics. I don't even know what those compounds are yet. And then we also did some early development tests, and we found a response in corn in terms of total root length. For the exudates, once again, soy and corn did not really induce a strong response, but with sorghum, we saw a reduction to the point of, to the same level of water. So basically, once again, like I showed you with the foxtail, the seeds that did germinate here developed quicker. On the same time period, they produced more roots. And it could be that they're trying to compete more. In terms of the shoot dry weight, we saw that the corn plants germinating in corn exudates were the biggest, and there was a response. We also saw an interesting response in soy where the shoots of plants, of seedlings in corn exudates of soy were the biggest. So maybe once they're realizing they're under competition, they carry things around, is what we're thinking. We also looked at shoot-root ratio, but for lack of time, I might not go into it. So now we're analyzing this data, and we're thinking about what we want to do next. So the literature now has 2 different opinions, and they're both equally as loud, if you want. So it could be that corn plants uptake more phosphorus by just sensing the existence of soy plants next to them. It's possible. It's been claimed in the literature. Other papers say, no, actually the exudates from soy plants make phosphorus more available, and that's why there's increased uptake. There's also a paper that showed that there's higher transcription of phosphorus transporters in the roots of corn plants grown with soy, but recently there's been a C correction thing added to that paper where if you look at it, it's not really happening. So I don't know. We're very curious about this because if we can cause plants to uptake more phosphorus just by putting in exudates, it's like a biostimulant. We just have to grow them in water and use that water in irrigation, and bang, we get more phosphorus in the corn plants. If that exists, it's amazing. Another thing we're thinking about, and I'll maybe just say that quickly and jump to my summary, is there is a claim that soy plants will fix more nitrogen when they're grown next to corn. To me, that is not evolutionary stable. There's no reason for a plant to be altruistic. It could be that they're expecting some nitrogen deficiencies later on, but these are not plants that have co-evolved for a very long time. So we're very curious about this, and this is what we're planning to do now. We're going to ask these questions. And the first thing we're doing is growing all three plants in a hydroponic solution with high nutrient availability to try and see what the roots do. We're later going to try and grow them in pots and see if we see elevated uptake, which I don't know what to expect. It's a very interesting situation where I do actually have a two-sided hypothesis. I do not know what to expect. The literature can point you in both directions. It's exciting times. But if any of it works, it's amazing. It's something that I'm very hopeful can lead to better outcomes for farmers. And then one last thing I wanted to say is that I recently partnered with the Botanical Society of America, for which I'm hosting a new podcast. And sorry, Jenny, I just realized you were on the last one too. And I think it's super, super important for us to present our science in a more approachable and easy to find way for the general public. I think in this day and age, and I was strictly warned by my advisors to not talk about politics, but I think we all do what we do because we believe it's true. And I think truth needs to be as easily accessible as misleading information. And this is my small attempt at doing that. This should be launched in the next month. And if any of you are working on anything related to plants and are either members or affiliated with the Botanical Society of America, reach out to me. I would love to have you on. It's a really fun experience. I'm fun to talk to on Zoom, and it's a really easy experience. And those are expected to have a big crowd. 1 o'clock. Yeah.
All right, so I think we can start with questions in the room.
I have a slide for that.
Yep.
Hi. So some research in our lab suggests that maize plants through different compounds as they develop. Did you play at all with the age of your maize or other plants in capturing their exudates?
Did not, am expecting to. And the reason for that is that I had the fortune of overlapping with Erika Lawrence-Paul, who is a big vegetative phase change expert. So we know it's true. We even know it from Striga. I agree with you 100%. We've not done that yet. It's something we're planning to do, but I couldn't agree more. There's definitely changes. Yeah.
Thanks for the presentation. I was wondering, so plant-plant interactions, I've done a little bit of work in like crop-weed interaction, same thing, right? Plant-plant.
Mhm.
Are you familiar with like Clarence Swanton's work from the University of Guelph?
I'm not.
Okay, well, it's interesting because I think it goes along nicely with this research where he also proposes that, yeah, like crop yields being limited by weeds. Yes, there's like a fertility and shading element, but even before any of that, he proposes that the primary thing that is limiting these yields are just like the sensing of somehow this weed or this crop is sensing that there is another plant in its vicinity. And I thought that was like, when you got into like exudates and allelopathy and also a lot of the weed biomass work that we do is like all above ground, but so much is happening underneath.
I fully agree. Yeah.
So it's like plant-plant interactions, but also I was like, yeah, crop-weed interaction, same kind of deal.
Yeah, and that also feeds into some work that's been done in Penn State with cover crops where they do mixtures, and those mixtures actually do better than monoculture. So maybe we should probably be looking deeper into this. Excuse the pun, but yeah, yes.
Thanks. Great talk. Yeah, so plant breeding is in part a numbers game. We have to measure as many plants as we can just to do selection. You mentioned idiotypes. I'm wondering if your research suggests some kind of intercropping idiotype that is faster to measure than, say, growing the intercrop and making a biomass cut and separating out the species, like that's slow. Is there something that comes out of your work that we could select on that would, by indirect selection, improve intercrop performance?
Oof, I have to think about it for a second. No, you're good. Well, for one thing, as a way of making the selection more high throughput, and I sort of touched on that with Greg, I think if we did hydroponics and we played around with the species in there, allowing for greater interaction and looking at the performance, it's not the field, it's very artificial, but it might give us a way forward, I think. So the way I had envisioned it is with the NFT system where the top plant is the exuding one and all of the rest are experiencing some sort of a gradient of exposure. I don't know. The thing we wanted to achieve with that sorghum work was we wanted to get RNA-seq data from before and after the exposure to the exudates in the exudate transfer thing. As life happens, we missed the deadline on one grant, and then my fellowship ran out. We did not end up doing it. I think we could see stuff there. I think if we did this in an environment that had multiple nutrients availability or varying nutrients availability, we can tease apart the receptors on the root that respond to that specific element, and that might give us a better target for breeding. I definitely think it's worthwhile. I've been hearing that we're running out of alleles for a very long time, and the root is maybe an untapped resource. I don't know, but I do think it's a possibility. Yeah.
Yeah. One question online. David, it looks like you have your hand raised. Did you have a question?
There. Yes. So this sounds like it's, so I work with Clarence Swanton quite a bit. And our hypothesis is that, the plants recognize each other, and that's why you get sort of reduced yield. And if that's the case, then it should be possible to identify mutants that don't respond to those chemicals, right? That just ignore them. Have you thought about looking for mutants that do not respond to the exudate chemicals?
I have not. We did hope that if we grew land races, we might see variation in the direction of response. We did see variation in the magnitude of the response to exudates, so there could be some differences there. It was a very small sample. It was 10 lines, and they were mixed between land races and elite lines, so it wasn't a really good panel to ask that question. I would imagine it's there. We've not done this. I will say that I looked at some strigolactone sensing mutants in peas, doing some other work, and I'm less convinced that strigolactones are the key agent in this sensing, but I could be wrong about this. And with sorghum, those mutants weren't readily available for us to use. It will be a logical next step, which we're hoping to achieve, grants becoming available again, permitting and such.
Excellent. Just so you know, we do have a lot of RNA-seq data online for plant weed interactions. You're welcome to mine it for whatever you'd like. So,
thank you.
All right. Any other questions in the room? I think that's it on. All right. So let's thank the speaker once again.
This has been a production of Cornell University, on the web at cornell.edu.